Right. So this diagram here is showing you the major um, components of your digestive tract. Obviously, you're expected to know a diagram like this, right? Now, in terms of the major functions of the digestive system, obviously ingestion, because you have to be able to move food into the digestive system. Of course, when you say ingestion, we are talking about the mouth. Then, of course, we have mechanical processing. Now, the first thing I want to notice, which will be a little different from what many of you were told at CSEC, is I have mechanical processing here. I don't have mechanical digestion. Okay? There's a reason for that. Mechanical digestion actually does not exist. It's not a real thing. Digestion requires chemical breakdown. Okay? So when you're chewing food, you're not digesting it. You're taking big chunks and turning them into small chunks. So you're processing the food for size. Okay? So remember now, no mechanical digestion, it's mechanical processing. And of course, mechanical processing occurs obviously in the mouth by the teeth, but it also occurs at several other places along the digestive tract. For example, it occurs in the stomach as well. Okay? And some amount of it also occurs in your small intestine. Chemical digestion, of course, this is now the real digestion. So this is taking big molecules, breaking them down to small molecules. And I'm sure everybody remembers from CSEC, the protein to polypeptides and polypeptides to amino acid business. Or we can say starch, right, to disaccharides and disaccharides to monosaccharides. Or of course, fats, lipids, fatty acid, and glycerol. This is stuff you all did at C6, so I'm sure you remember that stuff, right? We will be looking at that as well, but we're going to concentrate more on the enzyme side of things. So now after you have chemically digested food, you have to be able to, in order to chemically digest food, you must be able to add secretions to the food. So of course, secretions, that's when we're talking about enzymes, right? But in addition to enzymes, there are also other secretions that are required to allow the food to move easily through your digestive system. So mucus, for example, and we all know how important mucus is. It's all over the place, right? It's an example of something that will be secreted, right? Pancreatic fluid or pancreatic juice, if you prefer, right? As well. That does contain enzymes, of course, but there are other things in there. Then, of course, we have absorption. Simply put, once you've broken down your polymers to monomers, you then move those monomers from your digestive tract into your bloodstream, whether it's directly via capillaries or indirectly via the lymphatic system. Eventually, the nutrients will get into your bloodstream, right? And of course, the last bit here, whatever is left, whatever undigestible material is in their digestive system has to be removed. That, of course, is a job of digestion, right? So you should already be familiar with all of these nice, easy steps. That said, we're going to look at the general anatomy of the digestive system in a little bit more detail than you did at CSEC. Then we'll swing back around to the physiology, of course, which is represented by this, okay? So you already know from ANP1 that the abdominal pelvic cavity is lined, um, one second, please. You already know that the abdominal pelvic cavity is lined with a serosa because pretty much all the cavities are lined with serosa. And of course, our serosa is normally in two parts, a visceral part which is in direct contact with your organs in the abdominal pelvic cavity, and the parietal part of the serosa, which is in direct contact with the cavity itself. Now, the serosa in the digestive system is known as peritoneum. This is also something you'd have done in ANP1, so I know you know this one already, right? And of course, as we know, generally speaking, between the two layers of serosa, you always end up with a little cavity, right? You remember pleural cavity, for example, when we're doing the respiratory system. And within that cavity, just like in the respiratory system, you're going to have fluid produced for the purpose of lubrication. Okay? Now, that's why it says your peritoneal fluids line the cavity. Now, obviously, in the lungs, it was pleural fluid, which of course was important for lubrication. In your digestive system, which is lined with peritoneum, it is going to be peritoneal fluid. But functional is the same stuff. But of course, we had named it differently because it's in a different place, right? Then now we have the mesenteries. The mesenteries are sheets of membranous tissue that are needed to hang essentially the components of your digestive tract. It attaches them to your body wall. So when you're moving around, I don't want you to get the impression that your intestines, for example, are sitting in one big pile in your abdominal pelvic cavity. And as you're moving around, you're sloshing around all over the place. That's not the case. They are actually attached mainly to the body wall in some fashion or the other via these mesenteries, okay? So the mesenteries are designed to hold the various loops of intestine and stomach and rectum and other things in place, relatively speaking. I mean, it does hang, so there's some amount of movement that can be associated with it. But think of it, it's more along the line of think of fruit hanging from a tree, right? It can move a certain amount when the breeze blows, but it's not going to go very far. So yes, your intestines can slosh around a bit, but they're held in relatively stationary though. Okay. Now, the mesenteries themselves, as it says, they're a double sheet of peritoneal membrane, and the peritoneal membrane here allows blood vessels and lymphatic vessels and nerves to run alongside and to innervate your digestive tract, right? So it's not just there to hang your intestines and your stomach. It also provides access route. Okay.
So can you repeat back that part, please?
All right.
About
What
the
I'm
around.
saying is that the mesenteries, which are basically wrapped around the different sections of your intestines, will attach, and the digestive tract in general, will attach the tract to your body wall normally, right? In addition to attaching the different parts of your digestive tract to the body wall, mainly, we'll talk about the liver in a second. It also acts as a area where blood vessels and lymphatic vessels and nerves can run alongside the digestive tract without fearing damage. Okay? So they're running through the mesenteries. And then from there, like the blood vessels can spread into the walls of the intestine and the stomach, etc. Nerve tissue can do the same. Lymphatic tissue can do the same. Okay? So the mesenteries are just not just there to stabilize the intestines, they also allow for the other major tubes associated, of course, with life, right? Now, it also stabilizes the attachment points to organs. So, for example, your pancreas is attached to your small intestine, right? You're going to find the pancreatic well the ampulla is what is referred is what is called, which is a tube that runs to your small intestine, okay? That area there has to be anchored properly to your body wall because these are narrower tubes than the intestines. They're more susceptible to being damaged by twisting, okay? So the idea here is, of course, that in the areas where you're joining other organs to the digestive system, you need to have the mesentery stabilize these areas because those are areas of potential failure. They're easier to damage than other places. So they need to be properly secured, right? Okay. So this is just this diagram here, and this one are pretty much the same. Of course, this one is bigger, so it's easier for us to point out the bits and pieces. So what I want to point it to you here is your liver, of course, is an accessory organ of the digestive system. And you'll notice here the liver is attached to the diaphragm via what we call a falciform ligament, okay? But the liver itself also is attached via the sheet of tissue here called the lesser omentum, and it is attached to the stomach. So the stomach is partially hanging off of the liver because of this bit of mesenteric tissue right here, right? Then when you go down further from the stomach, you see another sheet of mesentery that comes down down further into the abdomen before it folds up and then it surrounds, of course, sections of your intestines. Okay, we call this sheet here that runs down the greater omentum, and then of course from the greater and lesser omentum we get what is referred to now as our mesentery proper, where you have the mesentery surrounding various sections of the small and large intestine. And you can see here where they're all attached to the body wall right here. Okay. So hopefully this diagram shows you the idea that yes, your organs are hanging and then yes, they're attached. And the points of attachment allow for some movement, but not enough for the organs to damage themselves. Places where you have accessory organs meeting the digestive tract are further reinforced with tissue to keep them as stable as possible. So when you're running and jumping and falling over, you don't have to worry about your intestines twisting up. Right? They're suspended essentially and held reasonably in place. Right. So this shows you here your mesenteries, and as you can see, here are your blood vessels here running through the mesentery tissues. So the mesenteries are not just there to suspend your small intestine, large intestine. They also allow for access via vessels, right? So as I said, mesenteries, digestive tract, accessory organs, they're suspended by dorsal and ventral mesenteries. It mentions here in embryonic life, right? Then now the ventral mesenteries are going to become the lesser omentum, which I mentioned earlier. Remember, the lesser omentum is going to attach now to the liver. Okay, so this here, this is our lesser omentum, and of course it attaches to the liver, and then the liver attaches to the diaphragm. So the liver the stomach is suspended essentially, partially from the liver. Okay, then now coming off of the stomach itself, we have this sheet here of tissue, and this is now the greater omentum. Obviously, this is from the anterior view, right? And this one is going to cover your intestines, right? So it comes right over like an apron and covers the intestines, large and small. So that's why it mentions the dorsal mesentery becomes the greater omentum, whereas the ventral mesentery becomes the lesser omentum. And of course, we mentioned the falciform ligament already, right? All right. So mesentery is pretty straightforward. There's not a lot there besides that for you to really harass yourself too much about, right? It should be fairly easy. Just remember, greater momentum, lesser momentum, where they come from, where they're going. Remember, mesentery is proper, and the functions of the mesentery proper, which of course the stabilize the large intestine, small intestine, some accessory organs, etc., and you'll be fine, right? Then now we can look now at the histology of your digestive tract. The good thing about the histology of the digestive tract is that just like for most of the respiratory tract, the layers are basically the same throughout. By that I mean, the innermost layer is always a mucosa. Then the layer after the mucosa is always a submucosa. And then from there, you always get muscularis. One of the differences is that sometimes you have a serosa, and other times you have another structure called adventita. And we'll talk about the difference in a minute, okay? But for the most part, these are the layers. Whether you're talking about a section through the esophagus, small intestine, large intestine, it doesn't matter. These are going to be the layers. Now, obviously, there will be differences in the way the layers are formatted, right? But fundamentally, just remember the layers. In the innermost mucosa, then submucosa, then muscularis, then usually serosa. In some places, like the esophagus, it would be adventita. Okay? And we'll talk about it in a second. All right. So with that in mind, let's look at the major components of the mucosa. So these individual layers here are all a part of the mucosa. So epithelial tissue, as I'm sure you remember from ENP1, the epithelial tissue of your intestines is comprised mainly of simple columnar epithelium. All right. And you already should remember what simple columnar epithelium is, right? I'm going to assume. Then the reason for that, of course, is because simple columnar epithelium is good at absorption and secretion. Two very important components of your digestive tract. Below that, you have an area called the lamina propria, which tends to contain small glands, a few capillaries, things like that. Okay? Then below that, you have the muscularis mucosa. This is actually a thin layer of muscle that is used to contract and to help potentially squeeze down on the lamina propria to help to force out glandular secretions. It can also aid in the narrowing of your digestive tube. Okay? It's not the only thing it's for, but it doesn't do it all on its own, but it can help. Right? All right. That said, let's jump now to this and see if what I'm saying matches what this says. It mentions here epithelial layer. The layer is folded to facilitate large surface area. We'll talk more about that later when we're looking at individual sections. It mentions the epithelium may be simple or stratified. This is now looking at the, remember when I mentioned simple columnar, I was talking about the tubes here. So we're talking about large intestine, small intestine, right? But if you go back from the oral cavity, you're going to get stratified squamous, of course, because of course, as you're chewing, the food is abrading the inside of your cheeks. The pharynx, of course, will also contain stratified squamous because, of course, once again, swallowing, the food is still in reasonably large chunks and not properly processed, so it can also scrape the pharynx. And the esophagus will also have stratified squamous, right? As will the anal canal. Now, once you get into stomach, small intestine, large intestine, where of course absorption and secretion are going to be important, that is when you get your simple columnar epithelium. Then now from there, lamina propria, as it mentions here, contains some blood vessels, some nerve endings, lymphatic vessels as well. And there are a few scattered smooth muscle cells and a few lymphoid tissues as well. This, of course, is because your digestive tract on a whole is a major place for infection, right? It is a major route by which infection, pathogens in general, can make their way into your body. So it makes sense to have lymph nodes, which, as you know, are a part of your immune system, lining the digestive tract. It's important to have it, right? And glands, as I mentioned earlier, in lamina propria, you have several types of mucus secreting glands as well. Okay. And remember, the lamina propria is right below the epithelium. So the mucus, etc., can be secreted and passed through, right? Then now we have the muscularis mucosa. As I mentioned to you, this layer here, of course, being muscle, is going to contract. As I mentioned, it can put pressure on the lamina propria, which helps to squeeze out glandular secretions, and it can also help to partially constrict around the the the, the mucosa to help to narrow the lumen. Help to, right? Just help to. All right. Then now submucosa, if you go back here, submucosa, here are the major components. Submucosa tends to be fairly glandular tissue. So you will have submucosal glands. Most of these glands are going to be associated with producing not just mucus, but they can also produce various secretions that aid in digestion, right? You will also find your major blood vessels here, and you will even find a network of nerves called a submucosal plexus, okay? Which of course means you can send and receive signals from the submucosa, increasing activity, for example, of glandular secretion or decreasing glandular secretion as the need may be, okay? So let's then jump to this. As it mentions here, it's a layer of dense connective tissue, which of course lies below the mucosa. We already mentioned major blood vessels will be found in there, as well as major lymphatic vessels, glandular tissue. You will find exocrine glands. Remember now the difference between exocrine and endocrine glands. Endocrine glands have no ducts, and their secretions go straight into the blood, right, via tissue fluid, interstitial spaces, right? Exocrine glands always have ducts, and their secretions, which will vary now, they're not producing hormones. These secretions go onto a surface, whether it's an internal surface or an external surface. So your skin would be an external surface, but the lumen of your digestive system is considered to be an internal surface, and it is technically not a part of your body, it's a part of the outside, right? The lumen. And I'll talk more about that in a little bit, right? So exocrine glands in the submucosa can produce buffers as well as enzymes. Buffers, of course, are used to help to control changes in pH. Because as you know, enzymes are very sensitive to pH. So you want to, so for example, in your small intestine, enzymes in there work best at a pH of between 7 and 8. All right, closer to 8 than 7. But yeah, secretion, but of course, the chyme coming from the stomach is going to be very acidic. So of course, um, buffers, pancreatic juice, other things, bile, all of these things will come together to help to manage the pH of that part of the small intestine, right? Now, the submucosal plexus, as we mentioned, also known as the Meissner's plexus, is going to contain sensory neurons as well as parasympathetic ganglions and sympathetic ganglia. Now, remember, please, in the digestive system, and you would have done this in AMP1 again. See why you have to do AMP1 first? The parasympathetic nervous system increases the activity of the digestive system. So if you activate the parasympathetic ganglia, the submucosal plexus will cause exocrine glands to secrete more often, for example, right? Whereas if you activate the sympathetic postganglionic fibers, it reduces the activity level. So you produce less substances from these glands, right? And this,
Sir,
of
can you
course,
repeat again, please?
in the digestive system, parasympathetic speeds things up, sympathetic slows them down, right? Unlike the rest of the body. So if you activate the parasympathetic ganglia, you are going to increase the secretion rate in the submucosa. If you activate the sympathetic fibers, postganglionic fibers, you decrease the secretion rate of those same glands, right? So remember, parasympathetic speeds things up in the digestive system, sympathetic slows them down in the digestive system only, right? So this diagram here just shows you one of the nice big glands that you can find in the submucosa. And of course, glands have ducts. So here's an example of a duct right here, nice and simple, right? Now the muscularis externa. Let's jump back and see what are the layers. You'll notice that the muscularis externa has an inner layer of circular muscle, right? And then in some cases, you will then have a layer of myenteric nerve plexus. Then an outer layer of longitudinal muscle. Now, both of these muscle layers are, of course, going to be smooth muscle, right? The myenteric plexus, of course, is used to activate these muscles. Because remember, the myenteric plexus is just a nerve mesh. So you can increase the rate of contraction or decrease the rate of contraction of these muscles by activating. Once again, if you want to speed up contraction, you activate the parasympathetic nervous system. If you want to slow it down, you activate the sympathetic nervous system, right? The only other thing I would say is that these two muscle layers are ubiquitous, but in the stomach, you're also going to have a layer of muscle called oblique. And the oblique layer will be the first layer instead of the circular muscle, okay? But for the rest of the digestive tract, pretty much circular is the innermost, longitudinal is the outermost layer of muscle, right? And you'll be fine. So, muscularis externa, as it mentions here, smooth muscle cells, forms inner circular and outer longitudinal muscle layers, as we mentioned, and contraction facilitates peristalsis and segmentation. Now, peristalsis, I know you guys are familiar with peristalsis. Peristalsis, of course, is simple. It is the muscular contraction which leads to the propulsion of food down the digestive tract, right? That should sound very familiar to everybody. You would have done it at CSEC, right? So peristalsis moves food from the mouth to the anus, basically in one direction, right? Segmentation you may not have heard of. Segmentation is a little different. Segmentation is, all right, here's the easiest way for me to describe segmentation. You ever bought like a box juice, like a box orange juice or one of those, or even one of these bottle, quote unquote, natural juices or real juices, even though most of them are concentrate, but still. And on the label, it will say shake well before use, right? When you're shaking, what are you doing? You're mixing up the particles in the juice to form a more homogeneous suspension before you drink it, right? You're mixing things up. You're homogenizing the liquid. In segmentation, that's pretty much what you're doing. You're taking the a bolus. You all know bolus from CSEC, which is basically the food that you swallowed moving to the digestive system. Okay, what you do is you take this bolus, and then what you do is you secrete enzymes onto the bolus. Okay, so you can see here So you can say, here's a bolus, here's some enzymes. So you then mix them together, and then you move the bolus and the enzyme mixture pretty much like this. You're shaking it up. You're pushing the bolus forward and pushing it back. Pushing it forward and pushing it back, right? So back forth, you're mixing up the bolus with the enzymes, with the mucus, you're mixing everything up to make a more homogeneous um, bolus. And why are you doing that? Because if you mix the enzymes in properly, you speed up the rate of digestion. If you mix up the buffers properly into the bolus, let's say it's coming, it's chyme coming from the stomach, you help to moderate the pH for the more efficiently. So segmentation, literally, is moving the bolus back and forth, and mixing substances into it. So you're essentially mixing with this back and forth movement, just like it's shaking up a box of juice, right? Whereas segmentation, remember, you're only moving substances in one direction, only from the mouth to the anus. As you can see here, it gives you the impression that peristalsis is like squeezing toothpaste out of a tube. You squeeze the base of it, the toothpaste goes forward, right? So basically what you have happening here is circular muscle behind the bolus is constricting, pushing the bolus forward. At the same time, longitudinal muscles are contracting, shortening the area in front of the bolus, making it easier for the bolus to move. Okay? Now you did this at C-sec, so hopefully that you remember that stuff, right? So this is just showing you here the inner circular muscle layer here, outer longitudinal muscle layer here. But if you look carefully, you can see this layer right here between them. That there is the myenteric plexus. So make sure if you see a diagram or an image, because test 2 is going to have a fair number of images on it, that you can differentiate the different components. And obviously, test 2 will be heavily skewed towards urinary system and digestive system, right? All right, so muscularis externa, it mentions here, they have an enteric nervous system. Remember the myenteric plexus, right? Which coordinates movements of the muscle of the muscularis externa and it's innervated by the autonomic nervous system. Now, the reason they mention enteric nervous system here separate is because the enteric nervous system can act independently of your general nervous system. The enteric nervous system does not need instructions from your brain to tell the muscles to contract, tell the muscles to relax. No, the enteric nervous system is getting information from receptors within the walls of your digestive tract. And based on these receptors, the enteric nervous system can tell the muscles to contract or relax, right? So you can think of, for example, pressure receptors, stretch receptors in your digestive system. As food is pushed into the digestive tract, you're stretching the area where you're pushing the food into, right? So if you go back here, you're stretching the area where the food is pushed. This, of course, stretch receptors pick this up and feed this information back into the enteric nervous system. And the enteric nervous system now will activate the circular muscle and longitudinal muscle, which helps to push the bolus along. You do not need the input from the medulla oblongata or any other part of the brain for this to function, right? Which is actually very important. Why is that important? See if this makes sense to you. Somebody who's a quadriplegic, right? They've broken their neck, right? Fundamentally, or there are no more signals traveling from their neck to the rest of their body. How do they stay alive? They can still swallow food. So even and the food can still pass through the digestive tract. Those muscles there, those smooth muscles, because they are in them are somewhat independent of your brain, they don't need to be in contact with the brains. Even if the spinal cord is damaged, they keep working, right? And if it wasn't for that, you can imagine paraplegics and quadriplegics would have a far worse life if they could have a life at all, right? So yeah, so enteric nervous system being independent is important. Now, as you mentioned, now, but with sympathetic fibers, it mentions your sympathetic fibers will also innervate the mucosa and the myenteric plexus. So yes, your brain can send information, right? And the plexi will respond to information from your brain. It just doesn't need it, right? We already know what I showed you the myenteric plexus already. And as we know, parasympathetic muscle stimulation is going to increase muscle tone and muscle activity in your digestive system. All right. So serosa now is the outermost layer, connective tissue mainly. So there's collagen in there, as well as elastin. It mentions it is found along most portions of the digestive tract, except for around the oral cavity, pharynx, esophagus, and the rectum. Okay? These areas here, you're not going to have serosa. You're going to have adventita instead. And the area around the adventita is dense collagen fibers, which attaches the digestive tract to surrounding tissue, right? Which helps to stabilize them. If you think about it, your pharynx in your throat, it has to be attached to the surrounding tissue so that when you're swallowing, those changes in pressure don't cause the pharynx to shift position. So you have to anchor it strongly in place. So a serosa is on the tube, right? It's sitting on it. The serosa doesn't necessarily anchor the digestive tract onto anything, right? That's what the mesenteries are for. The adventita is an anchoring tissue. Even the rectum, if you think about the rectum, when you're defecating, when you're trying to void your bowels, the rectal muscles have to squeeze down to push out the feces, right? So of course that causes pressure changes. Folks, give me one quick second here. Nice, Andrew. But I sent you the paper. But don't you have the cover sheet? No, you have it. I left it up there. Yeah, man, the day, remember? Yeah. Remember I said you keep the cover sheet because I don't need it. Check and see because I remember distinctly leaving it up there. I don't normally take them. EMP2. So you're good otherwise though. So you don't need anything else from me. All right then, baby. All right, sorry about that. That was some issues with your final exam. All right, all right. Let's do four more slides and I send you on the way, okay? So, sir, as I mentioned to you, the serosa is wrapped around the section of the digestive tract you find it, right? And let me just jump back here. And because, of course, you have the mesenteries surrounding the digestive tract, and these parts of the digestive tract, and these are attached now to the body wall, then this you don't need any further attachment. Okay. So in areas where you have mesentery, you don't need serosa, adventita, sorry. Now in areas like the pharynx, like the rectum, where you're not directly being attached via mesenteries, you need another way of attaching, which is what, of course, the adventita is for. Okay. So that the adventita helps to hold these particular places tightly in position. All right. Hopefully that makes sense, right? Then now we're gonna look now at the digestive system and the functions, the basic functions, right? As I said, let me get four more slides and I send you on your way, okay? So of course, we already know the major components of the digestive tract. We already know the oral cavity is for insertion, ingestion, okay? But ingestion is not just a matter of putting food into your mouth. There's more things that occur. So it mentions here, in addition to doing that, you have analysis of material. What does that mean? It means when you put food in your mouth, you don't just chew and swallow. Your tongue probes the food. You push the food against the side of your cheeks. You move it around. It allows you to analyze the material. Is it tough? Is it soft? Is it mushy? Whatever it is, you can analyze food. Anybody ever do this into whether you do like a blind taste test and you close your eyes or something and you stick food in your mouth? One of the first things you do is you roll the food around inside your mouth to get an idea of the texture and shape of it, right? Then, of course, we have the mechanical processing with the teeth. The tongue also helps with mechanical processing. Obviously, the tongue can't bite food, but when food is soft enough, for example, when you're eating ice cream or rice or something, you normally just push the soft food up onto the hard palate and use your tongue to rub it out, to mush it right down to nothing, right? Then, of course, we have lubrication. Plenty of mucus is produced in your oral cavity, particularly when there's food in there and you're producing mucus is saliva, which we'll talk about later, right? There's even some amount of digestion occurring in your oral cavity. I know you know this one here. Everybody knows salivary amylase from CSEC, right? But you also have something called lingual lipase. Now, if you're paying attention, you know ASC refers to an enzyme normally, and LIP, of course, refers to lipids. So therefore, lingual, of course, as in tongue, is lipase that is produced from the under the tongue. Okay? This is the reason why when you're eating, let's say you're eating some fried chicken, it's all greasy, and you don't have any napkins, what do you normally do? You lick your fingers. Why are you licking your fingers? Because we know oil is not soluble in water. So why does licking your fingers help? Because of the presence of lingual lipase in your saliva. When you start eating food that is heavy and greasy, you start adding more lingual lipase to the saliva, to the fluids in the mouth, which is why after you finish eating a very greasy meal, your lips might feel greasy, but inside of your mouth does not feel greasy, right? Now, I believe you would have done salivary glands at AMP1 already, but let's just run through them real quick. You have the parotid salivary gland, which is the largest salivary gland, and its main function is to produce salivary amylase. You have your submandibular salivary gland, obviously submandibular because it's mandibular because it's below the mandible. And this one here can produce a buffer solution. It can produce mucin, which is sort of like a very watered down mucus, as well as salivary amylase too, right? So submandibular gland does multiple different things. Parotid is only doing salivary amylase, right? Then the sublingual now is good for mucus, okay? So obviously I'm mentioning what the different glands are secreting because it's gonna be important, right? All right. Now, why do you need buffers? Why does the submandibular gland give you buffers? The pH in your mouth actually also has to be controlled, it has to be maintained because your teeth, the enamel, the dentine of your teeth are essentially alkaline, right? They're very susceptible to acidic conditions. They will dissolve away. Now, you may not know this, but I'm sure you've probably figured it out. Toothpaste is alkaline, right? So while you're brushing your teeth and you're removing debris of or whatever you've eaten recently, you're also helping to neutralize any potential bacterial acids that would have been produced during the course of the day, the hours before you are or after you ate, whatever it is, right? So the buffers are there for a similar reason. They, as you're producing acid, as the bacteria are producing acid, acid is starting to wear down your teeth. The buffers are supposed to neutralize the acids so the pH of your mouth doesn't drop much below 7. Okay. Now, in some people, they underproduce saliva, right? And they get a condition called chronic dry mouth. Now, if you are a person who's underproducing saliva, here are some of the things that will happen. You will have a harder time swallowing because the food will be, there'll be less saliva to moisten the food before you swallow. There, you will tend to have bad breath because the saliva would contain buffers, which help to, and as well as lysozymes, which are enzymes designed to kill bacteria. And without these in a fair volume, bacteria will of course breed up in the mouth and produce noxious chemicals, right? You will have a hard time. You remember I mentioned to you about cleaning your fingers with greasy fingers? You'll have a hard time doing that as well. Okay. That said, that of course leads directly into our last slide, which is looking at the functions of saliva in the mouth. Obviously, I just mentioned some, so let's mention some more. It helps to keep oral surfaces clean, and remember why, right? It moistens and lubricates food. It's mostly water, so that makes sense. It aids in tasting. How does it do that? By dissolving food in water, you form an aqueous solution, which means it is easier for the molecules in the solution to move into the receptors on the tongue, right? Maybe you've noticed it, that it is harder to taste dry food. And you only start, even let's say a pack of crackers. Try this. Just take some dry crackers and just put the dry crackers on your tongue and see what you taste. It's gonna taste probably a little salty and that's about it, right? When you start to chew the crackers now, then is when you can pick up the, assuming it's good quality crackers, the butter taste and other things in there. But only after you start to chew it because that is when you form the you mix the crackers now, you hydrate the crackers, you cause the molecules in the crackers to start separating out and binding to receptors, right? And of course, we know salivary amylase helps to break down the carbohydrates, which is why when you chew starchy food, if you chew starchy food for a long time, it starts to taste sweet. Even bread. Take a slice of bread, and the longer you chew the bread, the sweeter the bread tastes because you're breaking down the starches in the bread, right? And of course, it helps saliva helps to maintain the calcium phosphate matrix of the teeth. Remember, bacteria produce acid that will wear the teeth down. You buffer the acid as well as produce the lysozymes to kill the bacteria, all of which helps to save your teeth. Right? All right, so this is information about the glands here, which I mentioned to you earlier. All right, this is now slide 27. All right, cool. So we're at 27 of 114. So we're gonna try to finish this up on Tuesday, right? Following Tuesday, of course, you're looking at your in-course test. And in about a week or so later, you're looking at your final exam. I already mentioned to everybody how the final exam will be structured in the Tuesday class last week. Chances are I'll mention it again in the Tuesday class coming, and I'd like to mention it at least twice. Remember, it's 80 questions. There will be no questions on reproduction, no questions on the endocrine system, right? Everything else will be present. The final exam will be worth 40% of the total grade. In-course test 2 will be worth 10% now. Your labs will now be worth 30%, okay? So I'm just saying it again to make sure everybody's still on the same page with me, right? All right, let me stop here. I will be reopening the tutorials over the weekend. Honestly, I'm swamped right now. There's so many things going on with the semester. But give me until the weekend and I'll try to figure something out, right? I'll reopen them and I will send the email about people who want me to post the grades on Moodle. As I said, I'm just going to use ID numbers for those who don't object and put them on Moodle. For those who object, send me an email and after I deal with everybody else, I'll deal with your grades too. Okay? Otherwise, folks, have a good rest of your day and I hope to
So
see you.
before you end the meeting, when everyone leaves, I can ask you a question, please.
Oh, you want to ask me a separate question? All right, hold on. Let me pause the video then.